TCA cycle is sort of like the engine of life, okay? We say that it's because this is the pathway used to convert carbon, like organic carbon, acetyl-CoA, into inorganic CO2. Okay, so we're going to sort of take out all of the electrons from acetyl-CoA, and then the carbons become fully oxidized into CO2, and then the electrons come out as an ADH. Okay, now we're going to focus on that. Okay, so from acetyl-CoA into carbon dioxide. Here's CO2, here's CO2. All right, so again, TCA cycle. TCA cycle stands for tricarboxylic acid cycle. Tricarboxylate, basically it's referring to citrate. Okay, it's referring to this guy, citrate, citric acid, right? So we a lot of times also call it the citric acid cycle, or we can call it Krebs cycle. Krebs cycle is named after the guy who discovered this. His name is Hans Krebs, right? And the function of TCA cycle is really the central pathway for recovering energy from several metabolic fuels, because we saw that you can get fatty acid to acetyl-CoA, we can get glucose to pyruvate to acetyl-CoA. You can also go from amino acid to acetyl-CoA. Okay, so a lot of things will converge to acetyl-CoA. Okay, so acetyl-CoA is sort of like the end product of a lot of these degradation pathways. This acetyl-CoA can then be oxidized completely to then generate NADH. So that's what this means. Okay, for recovering the energy. Okay, the energy is referring to energy stored in fatty acid, in glucose, in amino acids. Okay, and then also, this pathway is used for providing a lot of different intermediates for several anabolic pathways. Like last time we talked about how alpha-ketoglutarate is used to make glutamate. Okay, things like that. Okay, and what then happens is your NADH that you release, NADH and FADH2, can then be passed on to O2. That will do the subsequent oxidation, oxidative phosphorylation process that will generate a lot of ATP. Okay, so this is the TCA cycle. Now, so here I give you a simplified or a simpler view of TCA cycle. You start with acetyl-CoA, okay, and this guy is citrate. Citrate, a lot of times you can draw like this, if it's easier for you. Right, it's the same thing, right? Because OH is in the middle, and you have 1 carboxylate, 2 carboxylate, 3 carboxylate. Okay, so TCA, right? Tricarboxylic acid. Right? It's tricarboxylic acid, right? Because that's 3 acid groups, right? So you can draw like this, if it makes your life easier. Okay. First thing it's going to do is it's going to turn citrate, this guy, into this guy. This guy is called isocitrate. Okay. Basically, the only thing that happened was you moved the OH from the middle to a carbon on the right. Okay. Now, what you should notice is that isocitrate, this thing now has stereocenters, because you see how we label out the chirality here. Okay. Now it has 2 chirocenters. And then from isocitrate, what you can then do is you do a very simple oxidation. So you turn the hydroxyl group here, okay? You turn that guy into a keto group, okay? So you do an oxidation, which is why you get NADH out, right? So this is a oxidation reaction. So you get NADH. Before we lose the CO2 here, what you should notice is that now this guy becomes a alpha beta relative to this guy, okay? Relative to this guy, alpha, beta, and if the OH becomes carbonyl, then it's a beta keto acid, right? So beta keto acid, so then this can release as CO2. Okay, so this guy, leave. Okay, leave because it's beta keto acid. this reaction is very similar to which reaction that we talked about last lecture or the lecture before. There was one reaction that we talked about that also oxidized a OH into C double bond O, and then it somehow was then able to make it into a beta keto acid, and then the carboxylate leaves. Do you guys remember? You probably don't remember which enzyme, but can you guys tell me which pathway? Which pathway have we seen very similar reaction? That by oxidizing a alcohol in the molecule, you turn this molecule into a beta keto acid, which then allows the carboxylate to be removed as a CO2. Pentose phosphate pathway. That's exactly right. Okay, so in the pentose phosphate pathway, there was an enzyme that converts, C6 to C5, right? That one is doing pretty much the same thing here. Okay, so you're going to do a very simple oxidation, but yet after you do the oxidation, you generate a beta keto acid, and because beta keto acid is less stable, so beta keto acid will actually kick out CO2. Okay. Now, what you should then notice, okay, you get this guy is called alpha ketoglutarate. By the way, glutarate is without the keto. Okay, so if it's just glutarate, it's a 5-carbon diacid. Okay, and 5-carbon diacids are useful chemicals for producing plastics. So for us engineers, sometimes we try to generate glutarate, or glutaric acid. The next thing is you get alpha ketoglutarate into succinyl-CoA. So what happens is this keto group, okay, is this keto group. Okay, so then what happens is you kicked out this carboxylate. Okay, you kicked it out as CO2. Okay, it leaves. This reaction is very similar to which reaction that you have seen. This molecule is an alpha keto acid. Why? Because here's the acid, then this guy is alpha. Okay, that guy is alpha. So you do a alpha decarboxylation essentially. Okay, alpha keto acid decarboxylation. And the result is a acyl-CoA. It's similar to which enzyme? Pyruvate dehydrogenase that we just talked about. Okay? So this enzyme is similar to PDH. We'll see that in just a minute actually. Okay? Now, you go from 6 carbon, 6 carbon, 5 carbon, and all of a sudden now you have 4 carbon. Okay, so the 2 carbons that you had for acetyl-CoA went into CO2. Okay, so in the first 4 reactions of Tca cycle turns acetyl-CoA into CO2. Okay, you agree? In 1 step, 2 step, 3 step, 4 step, you turn acetyl-CoA, which is 2 carbons right here, okay, into 2 CO2 right here and right here. All right. Now, the next thing that you have to do is somehow regenerate sort of the precursor for this step, and that is going to be oxaloacetate. So you have to somehow convert succinyl-CoA that looks like that into that. Okay. Now, so if you compare these two structures, this guy and this guy, okay, compare these two. What changed? Right? What changed? CoA becomes an acid, okay, and somehow this carbon right here becomes a keto group, okay, and then this carbon, nothing changed, right, and the terminal, this carboxylate is still carboxylate, okay. So really 2 things changed. One is CoA becomes an acid, and somehow the alpha carbon here becomes a keto group. Okay, so basically 2 things happen. Now, so this is the other 4 steps of Tca cycle, is somehow to get succinyl-CoA back to oxaloacetate so that you have oxaloacetate to continue the citric acid cycle to make citrate again. Okay, and the first reaction is simply get rid of the coenzyme A. Okay, get rid of the CoA, make it into an acid. And recall that CoA, like acyl-CoAs, right? Those things are high energy thiol esters, okay? So thiol esters, when they do hydrolysis, they can generate energy that is equal to ATP, right? So in that process, we're going to sort of conserve the energy in the thiol ester bond, and we're going to generate ATP. But here it's a GTP, but same thing, okay? GTP, ATP, CTP, and TTP, pretty much they have the same amount of energy inside. They're just nucleotide triphosphates. Here you can actually provide one equivalent to ATP, basically. All right? And then you go down to succinate. Now, succinate, you have to somehow put an double bond O here. How do we do that? The first thing is we're going to turn this alkane thing, okay, this sigma bond into sigma plus pi bond, okay? So we're going to do an oxidation, okay? So here it's an oxidation reaction, okay? And we release FADH2. And we generate this thing called fumarate, okay? And fumarate, okay? Fumarate now has a double bond. Then you can do a hydration, okay? You can do a hydration here into malate. The reason this hydration can occur, okay, is because here, relative to this guy, alpha, beta, right? So when we talked about enolase, yeah. When we talked about enolase in glycolysis, remember we said that in order to sort of pull out an OH, it has to be on the beta carbon relative to a carbonyl. Remember that? When we talked about alpha beta elimination, that kind of stuff, right? So go back to like, I don't know, 3 lectures ago, all right? Take a look, all right? So here, whatever, in the end, you generate malate, okay? So you turn double bond and you do a hydration and it becomes malate, okay? Malic acid. Malate can, now this is OH, so you can do another oxidation and then it becomes oxaloacetate, okay? Oxaloacetate, sometimes I just call OAA. A lot of times you will see a shorthand OAA, which stands for oxaloacetic acid, okay, because really it's an acid, right? OAA. It's a very popular bioproduct these days. Succinate itself can be produced as a fermentation product, okay? So there's some organisms that will naturally produce succinate as the terminal product. And it's used for, primarily right now, it's used for food additives. Okay, so usually used as a food stabilizer, but what we can do is use this and we can make bioplastics out of succinate. The first reaction here, citrate synthase. Citrate synthase is a very interesting enzyme, okay? You have to appreciate biology for their ability to create carbon-carbon bonds, okay? Because you guys learn organic chemistry. Creating carbon-carbon bonds sometimes it's not necessarily easy. Okay, you should sort of know that. Biology is doing this very well. Okay, so here we're creating a carbon-carbon bond between this carbon and this carbon. Okay, so essentially you take this guy, you connect it to this guy, and you generate this because the CoA will leave. Okay, so CoA sort of leaves, and then if CoA leaves, this thing is sort of organic acid. Okay? So what you basically do is you connect this guy to the keto group of oxaloacetate and then you generate citrate. Right? And what you should notice is the delta G for this reaction is huge. Right? It's minus 32. So it turns out this reaction is highly regulated. Okay, it's one of the most regulated steps for Tca cycle because the energy is so large. Right? If you want to do Tca cycle, you turn this enzyme on. If you don't want to do Tca cycle, you turn this enzyme off. Simple. It's both the first enzyme and one of the most exergonic reaction of the pathway. Makes sense. So it makes sense that you would want to control this guy. Right? Turns out it's actually quite interesting. Citrate synthase will change conformation in response to substrate binding. Okay, so it has an induced fit model. So what happens is it will bind first the oxaloacetate, and once it binds oxaloacetate, then it will bind acetyl-CoA. Okay? So it sort of changes conformation once it fits its substrate. Okay? So this is the rate limiting step for TCA cycle, or the controlled step for TCA cycle. And once you have oxaloacetate binding to the enzyme, okay, so enzyme change conformation. Now, the first thing that you have to do, okay, so we're talking about mechanism now. The first thing you have to do is do a deprotonation of acetyl-CoA. Okay, so in order to do this carbon-carbon bond formation, obviously this guy is going to be a nucleophile. This thing being electrophile. Okay? So in order to do any bond forming, you obviously have to have a nucleophile and a electrophile. Okay? So first thing you have to do is somehow pull out the proton on the alpha carbon, and hopefully by now you should be able to explain to me why is the alpha carbon's proton deprotonated, like why can you deprotonate it? Right? 可以吗？可以解释齁，就是 alpha carbon 上面的 proton 为什么可以被拔掉？就是我们不常说，如果我们今天要做 reaction， 对不对？Usually 就是你有一个 nucleophile， 然后 nucleophile 会去 attack 一个 electrophile， 然后你就形成一个 covalent bond 嘛，对不对？那 citrate synthase 这边呢，就是你需要 generate 一个 nucleophile， 然后这个 nucleophile 呢会去 attack 这个 electrophile，OK？So 这边 nucleophile attack electrophile， 所以你要先 generate 出这个 nucleophile，right？ 我们要把这个 alpha carbon 变成 nucleophile，right？ 那怎么变？当然就是你把一个 proton 拔掉，那它这边 electron 就会全部到这个 carbon 身上嘛，对不对？那为什么 alpha carbon 可以被 拔 proton？Right？ 这个是 carbonyl， 这个是 alpha carbon， 对不对？ So 为什么 alpha carbon 的 proton 可以被拔掉？因为它有 resonance， 对不对？就是你一旦这个拔掉以后，它是 carbon ion 会变，就会变成这个东西，对吗？所以它会有一个 resonance structure。哎，那有 resonance structure 呢，这个东西就算 kind of maybe a little bit stable。哎，那这个东西如果OK stable 的话，那你就可以去弄它。Make sense? Kind of, right? So basically what happens is you will be able to deprotonate the alpha carbon, and then you will generate this enolate. Okay, remember this guy is called a enolate. If you have a proton here, enol, right? And because deprotonated, you call it enolate. Okay, so enolate. Because it has the ability to do resonance, and then you get to this thing. And also, the reason why you can do this is because you have a histidine here that will stabilize this oxygen such that you get a hydrogen bonding going on. So this will stabilize this intermediate. Because you have a, histidine there that can then stabilize the oxygen, the carbonyl oxygen, so that when you pull out the alpha proton, then what you can do is sort of pass the general electron in this direction. Once you have this enolate, what then can happen is, of course, your minus charge can collapse back down. Okay? Once it collapses back down, here, the electron here is going to do something. Of course, it can go and grab a proton, and then you regenerate acetyl-CoA. Right? Like, do you know what I'm saying? Maybe? Okay? So then, when the electron comes down, this guy can, of course, go back, or it can go up. Right? So if it go back, it's the same reaction, so it doesn't want to do that. Right? So it's going to attack this electrophile, and you form a carbon-carbon bond. Now, this thing is also stabilized because when you go from the enolate, attack the carbonyl center of oxaloacetate, and oxaloacetate here, you will generate a tetrahedral intermediate where this thing is actually, you can go from like an O minus here, and then that electron will grab a proton from the histidine. Histidine become deprotonated. Okay, and you should remember why histidine can be deprotonated. 它的那个 pH 在 near neutral， 所以 histidine 呢最常会出现在 enzyme 的 mechanism 里面。Why？ 因为它的 proton 可以一下被拔掉，一下被放上去，一下被拔掉，一下被放上去，所以 histidine 常常会出现在我们enzyme 的 catalysis 里面。OK， 就 histidine 很重要，就没事不要把它 delete 掉。OK， alright. So once you do that, you will get this little intermediate called the citrulyl-CoA, which is, this is actually an intermediate because it's sort of like it's a compound, right? It's not attached to any enzyme. Okay, but, okay, 来咯，很重要哦，你看咯。From this to this, and from this to this, everything's reversible. Yes? Okay? So if it's reversible, you have no driving force to make citrate. Okay, so then what is driving the formation of citrate? It's essentially this. Okay, you're going to sort of do the hydrolysis of the high energy thioester citrulyl-CoA that makes this reaction irreversible. Okay, it turns out it's the hydrolysis of the coenzyme A, okay, CoA, that will, this step has a delta G less than 0, very large, such that this reaction is one direction. Okay, one direction. And all the other reactions were reversible. Okay? All the previous mechanism here, whether it's the deprotonation of the alpha carbon or the nucleophilic attack or the going from the enolate, the nucleophilic attack of the enolate to oxaloacetate, all of that were all reversible. Okay, they were all reversible. But when you get to the hydrolysis of the thioester, then it becomes irreversible. Okay, good. All right, so that's the first reaction. Okay, so energy very large. So the second reaction, aconitase here. Aconitase is a very weird enzyme. So what you're doing is you turn citrate into isocitrate. So if you only look at the substrate and the product, it's going to look like a isomerase. Okay, but turns out it's not. It's actually removing water and putting water back. Okay, so it's going to do its reaction in two steps. Okay, first step, you remove the water, basically here, and then you generate this CC double bond here, this intermediate called cis-aconitate, and then aconitase will then take water again and then will hydrate on another carbon, okay, a terminal carbon. All right, this thing is called the aconitase, okay, aconitase. Okay, so it turns citrate to isocitrate, okay? Now, turns out this reaction is very interesting because if we draw it like this, like sometimes we draw it like that, we will always draw isocitrate like this, okay? How come the oxygen is not here? Like, why not? Okay, because this thing, initially there's no chiral center here, but then why is it that the OH always ends up here? Okay, how come it cannot end up here? Or are they the same thing? Okay? So it turns out if you would start by radio-labeling your acetyl-CoA group, let's say you label your acetate, let's say we feed acetate and acetate becomes acetyl-CoA, and you label the acetate at the carboxylic acid part, okay? You do that and the bond forms between these two, so you generate a label citrate, okay? Now, you do aconitase, so you're going to change the OH onto a different carbon, okay? And you can hypothetically, let's say hypothetically, you can have two products, okay? If this guy goes to here, okay, then you have this. Okay. If this guy goes to here, then you have this. Right? Does that make sense? Right, where the O goes? Now, turns out we always only see this product. Okay? Why is that? And that is because aconitase can distinguish between the pro-R and the pro-S substituents of citrate. Okay, so citrate in the middle, you have one carboxylic acid right here, and you have another carboxylic acid, you have another carboxylic acid. Okay, so you have 1, 2, and then sort of 3, right? But if we look, the pro-S, this part and this part is a CH2COO minus, right? So it's 2 carbon carboxylate. And essentially what happens is your enzyme has a special organization such that it has to fit like this. Okay? And your reaction will always, okay, the OH is always added to this carbon, okay, to essentially this particular C. Okay, it's always added to there. Why? Because you can imagine your enzyme's active site is on the enzyme, right? This is the enzyme. Your active site is on the enzyme. So when your substrate, citrate, this guy is citrate, when citrate comes in, it has to point to the enzyme, not point up. Okay, if it points up, it's not going to react. Okay, now, if you were to turn this tetrahedral ball-looking thing, if you rotate it, if you rotate this guy down, then your OH and carboxylate is going to switch places. Make sense? If you like do a rotation. Yeah? Okay? So this is how this enzyme is always specific to generating the OH from essentially turning this OH onto here. Okay? Are we clear about aconitase? Okay, so aconitase is very specific. It will always turn the OH here onto the pro-R carbon. Okay? Obviously, Biochem 2 is one of those things where like metabolism it's kind of a lie. You have to remember a lot of stuff. But if you have to choose something to study, I'd rather you study the pathways. Okay? For example, what goes to what kind of, does it generate NADH? Does it generate ATP? Which step is probably the most well-regulated step in the pathway. For example, we talked about Tca cycle. The first reaction is going to be heavily regulated because the delta G is so large, right? So I'd rather you focus on that, but just throughout my class, I do have to talk about mechanism, but that's just how it is. It's a chemistry class, so we do have to talk a little bit about the mechanism, because otherwise, like in the future, if you end up doing, for example, designing, designing drug targets or designing some enzyme inhibitors, maybe these mechanisms will come in handy, so like how this molecule binds and whatever, that kind of stuff. Okay, so this knowledge is useful. It's not just me trying to bullshit you with a bunch of useless information. So we're good with reaction 1 and 2. Reaction 3 is this thing called the Isocitrate Dehydrogenase. Obviously, isocitrate dehydrogenase, like I said, it's a simple reaction where it's going to turn this guy into a keto group. So basically, this is going to leave as a hydride, okay, as a hydride onto an ADP, okay? So the way it works is very simple. Here you have a manganese cofactor that is used to stabilize your carboxylates. Okay, sort of it serves as a electron sink again. It sort of draws the electrons towards this direction. Okay, because of that, after you moved this guy as a H minus to an ADP, this one's gone. This is base pulled away. So you generate this keto group and you generate this Succinate. Okay, so this is like an intermediate. And remember I said this is alpha. This guy is then beta. It's a beta keto acid. So beta keto acid is relatively unstable because the product is relatively stable, so that the carboxylate will get removed. Okay? Like this. The carboxylate gets removed and you generate this enolate. Again, this is an enolate. And then that will grab a proton. Okay? Does that make sense? Once you grab the proton like this, you will generate your alpha ketoglutarate. Okay, so again, glutarate is 1, 2, 3, 4, 5 carbon dicarboxylic acid. Okay, that would be glutarate. And here is keto. Why is alpha? It's on the alpha carbon, beta, gamma. I forgot how to write delta. Delta? Whatever. Okay. Just kidding. Erase. Okay. So it's on the alpha carbon, so it's alpha ketoglutarate. Okay? So alpha ketoglutarate. And then next you have this alpha ketoglutarate dehydrogenase. Again, it's a complex, and you will notice that it uses pretty much the same cofactors as pyruvate dehydrogenase, where you will get rid of the carboxylate next to the keto group. Okay, so this is the alpha carbonyl. This is alpha carbon. So you're doing an alpha decarboxylation, which then you will then generate succinyl-CoA plus CO2. What you should also notice is the energy is also pretty large. Okay, why? Because it's similar to pyruvate dehydrogenase, and again, you should remember that pyruvate dehydrogenase has a very large negative delta G, and so this reaction is also irreversible. Okay, now, pretty much most, I wouldn't say all, but like most decarboxylation reactions are going to be irreversible because CO2 leaves the system as a gas, so it's harder to recapture the gas. Make sense? Okay. So it's very similar to your PDH. So here, this is PDH that we saw earlier today. You have E1, E2, E3. So alpha ketoglutarate dehydrogenase, so the alpha KGDH. Okay. Same thing. You have a very similar E2, very similar E1, and turns out identical E3. Okay, so in E. coli, for example, E3 is shared between alpha ketoglutarate dehydrogenase and pyruvate dehydrogenase. Okay, so it's actually this one is actually the same enzyme, but these guys have to be different. Okay, now you should think about why. Okay, you should think about why do I say that E3 can be the same enzyme, whereas E1 and E2, they have their own enzyme. Okay, think about that. 你觉得 PDH 跟这个 alpha KGDH pyruvate dehydrogenase 跟 alpha ketoglutarate dehydrogenase 它们差在哪里？差在 substrate， 谢谢，真的，它们差在 substrate 不一样嘛 ，OK？ 那 substrate 不一样的话，你觉得用同样的 enzyme 去 react OK 吗？不行嘛，对不对？为什么不行？因为 active site 可能形状不一样 ，right？ That makes sense。就是enzyme 我们不是说都很 specific to 它的 substrate 吗？合理嘛，对不对？那 alpha ketoglutarate 比 pyruvate 至少大快1倍，因为 pyruvate 是3个碳，然后 alpha kg 是5个碳嘛，对，所以大了 almost 快1倍。OK， 那为什么所以这个可以解释说为什么E1跟 E2 是有自己的 enzyme。OK， 那为什么E3可以用 the same enzyme？ 对，所以没错，所以E3呢做的一件事情是，它这边只是把那个 disulfide bond 有没有变回去？就它这边进来的时候是2个 thiol， 然后它要把这个东西2个 thiol 变回去 disulfide bond， 然后 electron 呢丢到 NADH 身上嘛，对不对？所以在E3的时候，已经跟你的 substrate 一点关系都没有了 ，right？ 它只是在做 electron 的 transfer，right？Right? So E1, E2 have their own enzyme subunits because the substrates are different in size, because one is pyruvate and one is alpha ketoglutarate. Makes sense. So they have to have different enzymes that have different shapes. Okay. E3, they can share the same core polypeptide, the same enzyme. That is because the function of E3 is only to return the 2 thiols into disulfide bonds. Okay. So the function for E3 does not involve pyruvate or alpha Kg, okay? It's aimed at only changing the thiols back to disulfide bond to your what do we call it? NADH. Okay, so E3 only does oxidation reduction, so it's not, you don't have to have a different enzyme for that. Right? Now, the fifth reaction, succinyl-CoA synthetase, okay, it's going to couple thioester hydrolysis with NTP synthesis. Now, in eukaryotes or like mammalian cells, we generate GTP, but like in, for example, E. coli, they generate ATP. It doesn't really matter. It's just a nucleotide triphosphate. So it's fairly simple. Okay, it's fairly simple. You will have a what? Some kind of nucleophilic attack. Okay, like this thing going to like fly over here, right? And this thing jump up, jump down, leave. Simple. Okay, and you generate this intermediate called the succinyl phosphate. Okay, so you actually change from a thioester to a phosphate ester. Then, okay, then what you have is on the enzyme you have a histidine, and the histidine will take the phosphate away, okay, through the nucleophilic attack on the histidine to the phosphorus, and you will kick out succinate. Okay, so succinate is kicked out, and then you are left with this 3-phosphohistidine. So it's just the high energy phosphate is now on histidine. So doesn't really matter where it is. The phosphate is still there. Then your GDP, okay, your GDP is going to come in, and it's going to attack the phosphorus through a nucleophilic attack. Okay, your PN bond is going to jump onto nitrogen, regenerate the histidine, and then you get GTP. All right, fairly simple, right? Maybe. Okay, so it's only a matter of, you know, transferring where the phosphate goes. Okay, the phosphate initially is in water, then it becomes on the succinate. Okay, become on the replace CoA and becomes succinyl phosphate, and then enzyme take that phosphate away, and you become histidine plus phosphate. Okay, so 3-phosphohistidine, and then 3-phosphohistidine then pass that phosphate onto GDP, which gives you GTP. This is like acetyl-CoA giving you acetate, same thing. Right? Now, this next part is then turning succinate into fumarate. Okay, and what happens here is you will kick out these H2. Okay, these one proton, okay, so 2 hydrogens and 1 pair of electrons. Okay, so you're going to leave with a hydride essentially. Okay, but not hydride, because you have 2. You're going to have essentially H2. Okay, hydrogen, essentially hydrogen. Okay, so like that. So your hydrogen is going to go from succinate to FAD, and then you generate FADH2. Now, there is a key characteristic that I want you guys to know. This enzyme is, FAD is tightly bound in the enzyme. Okay, so this is similar to when we saw pyruvate dehydrogenase, where FAD is in the enzyme. Okay, FAD is in the enzyme, so it's not flying around in the solution. Okay, it's not floating. Right, it's in the enzyme. Right. Fumarase is getting fumarate to malate, okay, and the way it works is where you go from fumarate and you're going to do a hydration, but using OH minus, okay, and you will get this carbanion ion transition state, and then you grab a proton, okay, then you balance it, and it becomes malate. Okay, so it's fairly simple this way, right? And then malate goes and become oxaloacetate through a oxidation where you're oxidizing this particular carbon. Okay, and again, this guy will leave as H minus to this one. Okay, and then you generate an ADH, and obviously this guy is lost as a proton. Okay, so you turn alcohol into carbonyl, so it's a oxidation, and because your carbon went through oxidation, you generate an ADH. So again, this is the overall pathway for Tca cycle. Okay. start with acetyl-CoA, and you form a bond between this carbon and which carbon? You have 1, 2, 3, 4. Your acetyl-CoA alpha carbon forms a new carbon-carbon bond with which carbon on oxaloacetate? 2号 carbon, because this guy is a keto group. Okay? Acetyl-CoA is going to attach to carbon 2. Okay? And then you generate this thing called citrate. Okay? So, on citrate, which carboxylate is your acetyl-CoA? 听懂我意思吗？就是你这边有3个 carboxylate， 对不对？这边哪一个 carboxylate 是从这个 acetyl-CoA 变过来的？可以是右边呢，也可以左边呢。对，就是不会是，总之不是它就对了。OK， 不是它就对了。OK， right? And then the rest is we talked about that. Alright, so if we look at the energy of Tca cycle, this is a chart that summarizes the standard energy, standard free energy change, delta G prime naught, and the physiological free energy change, delta G. Okay. Again, you should be able to explain to me what's the difference between standard energy and physiological free energy. Okay. Physiological free energy is essentially they are calculating the real delta G based on the concentration of the substrates that they measure inside the cell. Free energy 大家应该了解它是什么意思啦，我觉得 by now， 就是说你放1个 molar 的这个 substrate， 然后它多大的 energy 会变成 equilibrium 嘛 ，right？ 那是 free energy， right？ 但是 delta G 啊，这个 delta G 没有这个小圈圈，就是代表说它不是在 standard condition， 所以它的那个 substrate 跟 product 的浓度是有经过调整。什么意思叫经过调整？就是说我们其实可以去一个细胞把它打破以后，我们去量它里面所有的 metabolites。OK， 那我们是不是就可以得到一个浓度 of， 比如说 acetyl-CoA， 一个浓度 of citrate 这样，对不对？其实可以嘛， right？ 那样子的话。我们其实就可以去算出 the real delta G 大概会是多少 ，right? Make sense? So we can actually calculate the concentration of these different metabolites inside the cell, and then calculate the real delta G. So standard is actually pretty easy to calculate, right? Now the real is sometimes harder. Now, what you should notice is that you have a couple steps that have huge delta G changes. This one, this one, this one, and this one. Under physiological conditions, these guys we don't exactly know, but they're going to be negative, right? They're going to be negative because they're going to be controlled. So Tca cycle, you're going to control these 3 enzymes. Malate dehydrogenase. This one is going from malate to oxaloacetate. Produce an ADH. Delta G is plus 30. Then how come the real delta G is about 0? If I want to make a reaction or a conversion that has a standard energy of plus 30 to a delta G of around 0, what can I do? 提高浓度 of substrate and then 减少浓度 of products. OK， 所以我让 substrate 的浓度变很多 ，product 的浓度变很少 ，thermodynamics。OK， 用 equilibrium 去 push 它，就 OK， 可以吗？那 turns out oxaloacetate 在 cell 里面其实真的不会很多，因为 oxaloacetate 记不记得我说它同时是 alpha ketoacid 也是一个 beta ketoacid， 对不对？所以 oxaloacetate 本身自己就不是多 stable。OK， 那个我们去各位如果做生化实验哦，像我以前在做PhD 的时候。我那时候我有个反应我想要试 ，OK， 我要试一个反应，然后呢，我就可以跟那个化学厂商买 oxaloacetate，OK， OAA，oxaloacetate 泡在水里以后，然后你就开始看到它会一直产生泡泡 ，OK， 因为它会 spontaneously 变成 pyruvate， 然后吐 CO2， 所以其实 oxaloacetate 本身并不是一个多么稳定的 chemical，right？ 所以在细胞里面呢 ，oxaloacetate 通常 会很快速的被利用，就是像citrate synthase， 它会非常快速的去抓 oxaloacetate， 抓到 oxaloacetate 以后再去抓 acetyl-CoA，所以一切都 make sense. Okay? If we're talking about energy and regulation, what you should notice then is Tca cycle, you can understand Tca cycle as a process which it generates energy. Okay? It's not used to store energy, it's used to generate energy. So if your cell has a lot of energy, meaning it has a lot of ATP, it has a lot of like NADH, that kind of stuff, then it's not going to want to continue doing Tca cycle. Okay? So here are some regulators, okay, molecules that can regulate and control Tca cycle. For example, NADH. If your cell has a lot of NADH, it does not want to make more NADH, okay? Therefore, NADH is a direct product inhibitor of the NAD using dehydrogenases. For example, going from here and going from isocitrate to alpha Kg and alpha Kg to Suc-CoA. Also, you can also regulate this pathway through pathway intermediates where things like citrate and Succinate, okay, they can act as product inhibition or competitive feedback inhibition that turns off the Tca cycle. For example, if today you generate so much Succinate and Succinate is not fast enough to turn into oxaloacetate, then Succinate will come back and inhibit this guy. Okay? So that it's telling the cell, hey, stop doing Tca cycle. I don't need any more intermediates. I'm already full. Okay? And then if Tca cycle this is stopped, okay, then all of this will get stopped, and then you have citrate, which can feedback inhibit from oxaloacetate to citrate, and then you will not use acetyl-CoA. So also the level of substrate oxaloacetate and acetyl-CoA can determine the activity of citrate synthase. Okay, so turns out citrate synthase can actually be activated by acetyl-CoA and by oxaloacetate. Okay, we can turn on the activity. And also adenolates, adenolate meaning ATP and ADP, those, they should be fairly intuitive. If you're generating a bunch of ATP, then ATP will go and inhibit a bunch of reactions, right? Same thing for ADP, right? Okay, what's very interesting is that calcium, right? Calcium for animals, okay, is related to muscle contraction, and calcium is a allosteric activator for these dehydrogenases, okay? And that has to do with, let's say today you are in need of an ADH, and then you'll send calcium as sort of a signal to then allow the production of an ADH. Okay? So any questions on regulations of Tca cycle? The bottom line is you should understand the function of Tca cycle, and you should understand why they will be regulated based on these different regulators. Okay? Like ATP should regulate, that's obvious, right? Succinate-CoA could regulate, that's obvious, that kind of stuff. 懂我意思吗？就是 you know 你 sell 很多 ATP， 它就不想要 run Tca cycle 嘛 Okay? So it turns out Tca cycle intermediates are made and used in additional different metabolic pathways. For example, okay, citrate can make you cholesterol, can make you fatty acids, okay? Alpha-ketoglutarate used to make a bunch of amino acids. Succinate-CoA used to make this thing called porphyrin. We'll talk about porphyrin when we talk about secondary metabolites. Okay, so we'll talk about porphyrin. Malate can go back to glucose through gluconeogenesis. Oxaloacetate can make amino acids. Okay, and if you do today you are on a high protein diet, protein becomes amino acid. Amino acid can come into alpha Kg, can come into Suc-CoA, can come into Fumarate. Okay, so all of these things are interconnected. Okay, so that's really the center message that we need to get across from me to you at this third of the class. Okay, so central metabolism, glycolysis, Tca cycle, these things are all sort of interconnected. Okay, so they all can generate and produce things. Right? Now, there's one more thing then I need to sort of indicate to you, this thing called the catapularic reactions. Okay? These type of reactions are those that use cycle intermediates. Okay, so anything that consume Tca cycle intermediates, we generally call it a catapularic reaction. Things like making glucose, making amino acids, making cofactors, they will consume intermediates of Tca cycle. That means that we need to be able to replenish the intermediates of Tca cycle. And these are the endopleurotic reactions. Right? They will generate cycle intermediates from pyruvate, from amino acids, from odd-chain fatty acids. Okay, so those that generate are the endopleurotic. Those that take out are the catapularic. Make sense? Those are just keywords. Now, endopleurotic reactions are very, very important. Okay, Tca cycle being a cycle. So if you consume intermediates inside, okay, if you consume, for example, alpha Kg to make glutamate, okay, then you no longer have enough oxaloacetate to continue the cycle again, right? Make sense? 有听懂意思吗？就是说，你今天如果 cycle intermediate 被吃掉了，那你就没有办法回到 OAA 嘛，没有办法回去 oxaloacetate， 你就没有办法再做 Tca cycle， 就这么简单。Okay, so we really need to have a way to replenish Tca cycle intermediates, especially oxaloacetate. Okay, as long as you have oxaloacetate, then you can generate every other intermediate, right? Because if you don't have oxaloacetate, you cannot generate every other intermediate. Okay, so there are four reactions, four major endopleurotic reactions that are used to synthesize 3 synthesized oxaloacetate and 1 synthesized malolate. Okay, they're going to be from pyruvate and PEP. Okay, because pyruvate, there's a lot of pyruvate in you, and PEP, it's right before pyruvate, so there's quite a bunch of PEP. Okay, and these things are carboxylases, so what they'll do is they'll capture either CO2 or bicarbonate, okay, because these guys are C3 and these guys are C4. So you're doing carbon fixation. 你们有没有听过那个在生物学的时候，有没有以前高中或国中有没有听过那个植物有分 C3 plant 跟 C4 plant？ 有对不对？有这个东西对不对？那其中啊，它为什么是 C4 plant？ 就是因为它用到这种 reaction。从 C3 到 C4， 对，那这是一个方法来把这个二氧化碳CO2 带到植物里面。Okay, 所以其实就是靠这些 reactions. Now, first one, okay, this is the easiest one, PEP to oxaloacetate. Okay, PEP to oxaloacetate. You simply react it with a bicarbonate. Okay, you simply take a bicarbonate and then you get oxaloacetate. This thing is called PEP carboxylase. Okay. Another reaction is PEP, and what you can do is you can take in CO2 and then you also generate GTP or ATP, doesn't really matter. Okay. And then you generate oxaloacetate. Okay. Guess what? You saw this reaction before. Glycolysis and gluconeogenesis. Okay, so this thing is actually the reaction, one of the reactions for gluconeogenesis, going from oxaloacetate back to PEP. Same enzyme, but just the reverse reaction. Okay, why? It uses GTP and turns OAA to PEP. Right? This was found in gluconeogenesis. Right? Okay, but the reverse reaction also happens if you make it happen. There's also a type of enzyme called pyruvate carboxylase. Some organisms use this, like yeast I think use this. There's some bacteria also use this, where they can directly take pyruvate plus bicarbonate. Okay, so pretty similar to this guy. So very similar. However, here is pyruvate and here is PEP. So pyruvate does not have the same amount of energy as PEP. So it's going to use ATP as an energy source. Okay? And then you can use pyruvate to directly generate oxaloacetate. right? Or, okay, you can use pyruvate plus bicarbonate. Now here it's using NADPH, and you can generate malate. Okay, so this is called malic enzyme. Okay, so these 4 reactions you should know, okay, because they're very important for 2 reasons. 1, it replenishes Tca Cycle Intermediate, and 2, it can do carbon fixation. 你可以把 CO2 吃进来，这其实在生物里面很重要，真的很重要。没有很多 enzyme 可以做这件事情。Okay, sounds good. All right, there are some reactions that are very, very useful. For example, amino acid production, okay, using Tca Cycle Intermediates. We will get back to this when we talk about amino acid metabolism. Now, for example, you can go from alpha Kg to glutamate. I keep on saying that. And this is called reductive amination, okay, because we're not doing just, because here we're turning keto into amino, okay? So we're also putting an electron here, okay, a pair of hydride here, so that this is called the reductive amination, right? Or we can also do this thing called transamination, where we can sort of swap a keto group with a amino group. Okay, we can just swap them such that we end up with a amine on the oxaloacetate and becomes aspartate, and alanine becomes pyruvate. Okay, so you sort of swap amino group with keto group. And this is very useful because you can do a bunch of different amino-containing molecules, and some of them are used for drug precursor. So it turns out the production of pyruvate will actually increase the flux through Tca cycle by increasing the, what we call the substrate concentration. Okay, so the action of pyruvate dehydrogenase complex will actually increase acetyl-CoA pool, as does fatty acid oxidation. Okay, now oxaloacetate can limit that flux, but basically what this means is pyruvate is sort of the branch point between the endoproteolytic reactions to oxaloacetate and to acetyl-CoA. So pyruvate pool is then very important for Tca cycle. Okay, does that make sense, right? Right. So today, if you have a bunch of fatty acid degradation comes in to make acetyl-CoA, okay, just by having acetyl-CoA is not enough. You need to have oxaloacetate. And where does oxaloacetate come from? Okay, it comes from pyruvate. So pyruvate is sort of a very important control to the entire Tca cycle. Okay, but whatever. This is not super important. But you should understand how pyruvate has 2 functions, both providing flux to acetyl-CoA and for providing Tca cycle intermediates. Okay? Acetyl-CoA, obviously, if you have a bunch of acetyl-CoA, what it's going to do is it will inhibit pyruvate dehydrogenase, okay, and it will activate pyruvate carboxylase. 如果你今天吃一堆 fatty acid 进来，然后你这个 fatty acid 变成了 acetyl-CoA， 那问题说你有 acetyl-CoA 很多 ，OAA 不够，那你怎么办？你需要 OAA 啊，所以这个时候 acetyl-CoA 呢会变成是一个 regulator， 它会去把 PDH 关掉，就说我不要再做，就是不要再把 pyruvate 变成 acetyl-CoA 了，请你把 pyruvate 变成 OAA 好不好？所以它会去把 PDH 关掉，然后去把 pyruvate 到 OAA 的径打开。Alright, I promise this is the last thing. Carbon labeling. You have to develop a good sense of where these carbon end up. Okay? Let's say I put a radioactive label, like a C14 or C13 label on the methyl carbon of acetyl-CoA, like on here. And where is it going to go? Okay, so throughout Tca cycle, where does that carbon go? After binding with OAA, it's going to be right here. Okay? Or right here, but it's right here. Okay, so if we look at it that way, I mean it's going to be like binding right here. Right? So, and once you get citrate, isocitrate, alpha Kg, makes sense, right? Still here. Succinate-CoA, makes sense. We're good. All right, so far so good, right? Succinate, right? We're good. But what's wrong with succinate? Succinate is symmetrical. So C C3 or C2 and C3 essentially can be the same thing. Okay? Sorry, I should draw right here too. You cannot differentiate this carbon from this carbon. Okay, your enzyme just cannot. Unlike aconitase, okay? Your enzyme does not differentiate C2 and C3 of succinate. Because of that, when you go from labeled succinate, okay, to labeled fumarate, both of these carbon will be labeled. Okay? And when you go to fumarate to malate, both of these carbon will be labeled. You go to malate to oxaloacetate, both of these carbon will be labeled. Make sense? 懂我意思吗？就是说 acetyl-CoA 进来的时候是没有问题，到这边都没有问题。什么是左边，什么是右边很清楚。但是当你从 succinyl-CoA 到 succinate 的时候，你的左边跟右边已经分不出来了，因为它这个东西是 symmetrical。Okay? 所以 C2 跟 C3 cannot be differentiated. As a result, your enzyme when it reacts succinate to fumarate, 你的 C2 跟 C3 都会被 label， 就都会有。就是你拿出来的时候，就是你的 sample 里面，你去 measure 你的 fumarate， 你就会有 C2 被 label 的 fumarate， 也会有 C3 被 label 的 fumarate。Okay? 因为 fumarate 本身也是 symmetrical。Okay? 然后你到 malate 的时候更是如此，你这个时候就会有一个这边这个 carbon 带 OH 的会被 label， 旁边的也会被 label。Okay? 到 oxaloacetate 也是，这边会被 label， 这边也会被 label。Okay? So why do I need to tell you this? Okay? Why do I need to tell you this? The reason is because sometimes we want to know, for example, sometimes we want to know where does your particular carbon end up. Okay? This is a very useful method for guessing and elucidating the metabolic pathway. Okay? We need to know, for example, you feed acetate and then where does that acetate go? Okay? So this is very useful for metabolomics. If you want to study, for example, the difference between like a metabolism of a normal cell with a metabolism of a sick cell, you can do labels. Okay, you put labels and then you let it run. Okay, and then you have a bunch of different labeled metabolites, and then you analyze that. That will tell you what metabolic pathways are changed and what is not. Okay, so carbon labeling 是一个还蛮 important 的一种 experiment. Okay.